I think he's right. By the end of this presentation, though, the question is not whether or not or where it was made. The question was why was it made? Now, I'll prepare you. This is going to be just my thoughts based on years of observing. It's not corroborated with anything. It's just a few thoughts I've put together based on the science. But let's start first with what it is that Boris Johnson has said. In case you're not aware, Boris Johnson, the ex Prime Minister of the United Kingdom, is the most senior person to come out and say something like this. Now, it has been said by other agencies in the US. It was in a minority Senate hearing as well, where they raised this possibility, but it has largely been rejected by the scientific community that this could have come out of a lab. As far as they're concerned, it was in a wet market. They don't know which animal it was. They don't know how it got there. They don't know why the other animals aren't infected, but it was from a wet market. So, in order to understand this, sometimes I think a story helps. And I look at this story similar to the woman is inside the house. She's been stabbed. She died. The police come and investigate. They can find no signs of forced entry. There are no broken windows. There's no glass on the floor. The only person around was her husband. He says he didn't do it, but his fingerprint is on the knife. He said the only reason it got there was because he used the knife. He doesn't know who did this, but it's not him. He just so happens that this husband also gets insurance policy for 2 million. Do you believe him? If you do, you'd make a terrible detective. The point is that whether or not you believe him doesn't mean that you don't do thorough investigations. And that's what didn't happen in this situation. But let's just go straight to this story, which was in the Daily Mail on Sunday, the 29th of September. So that's today. And what it says here is that Boris Johnson is convinced that COVID was made in a Chinese lab. And what you have to realize is this is an explosive memoir. So he is selling a book. He rejects the theory it started in the Wuhan market and said it's likely the virus was result of a botched experiment. The critical thing there about botched experiment is extremely important. I will come back to that in a little bit. But you have to realize he has no longer prime minister. He has done a memoir, he has a book, and he wants people to read it. And one of the big things that he said in that is that the virus came from a lab. And it goes on to say that it now looks overwhelmingly likely that the mutation was the result of some botched experiment in a Chinese lab. Then going down here, there's a picture of Boris, and you have here Mr. Johnson is the most high profile world leader since Donald Trump to publicly reject the notion that the virus was transmitted zoonotically from infected animals. You know, in today's extracts from Unleashed. So this is his memoir, which is being serialized exclusively in the Daily Mail and the Mail on Sunday. So that's really what this is about. But it's still an important point that he said that he doesn't believe that it came from a wet market. The problem is that people seem to think that this is just an interesting point. And I keep on reminding people that if you don't go back to that original question, nothing is going to make sense before this and after this, because this is not over. The first mistake is that the only thing Chinese about this is that it happened in China. But this was a global collaboration. Very important to note. So don't think that this is about the Chinese trying to. No, no, no. This was a global collaboration, and you have to remember that Western scientists went out of their way to kill the possibility of you linking this to a lab. So there are vested interests, and the vested interest is that this research was being funded by international parties, and they did their best to keep it hidden. But it was exposed. And so, therefore, this is now more acknowledged that, oh my goodness, we were funding this. We didn't realize it was so serious. We didn't know what they were doing, and so on and so on. It goes. Why was it that I am not surprised? Because I had looked at the evidence at the time and I couldn't find any animal link. And it occurred within a mile of a lab where they do experiments on coronaviruses. I mean, that to me was just a no brainer. Why would you think anything different about the possibility? And then on top of that, I happen to know one of my research colleagues was part of a collaboration with regards to some unusual patterns about the spike protein that was on this virus. I'm going to show a short clip. I've done this before. This was part of a promotion that we did for a conference. This is over 2 years ago. So I've known about this for a long time. But I'll let you listen to this and you can and then I'll explain to you what it means. In the middle of 2020, just after the pandemic had spread across the world, a scientist who was trying to understand the virus and how easily it spread was looking carefully at the spike protein on the virus. There was an unusual pattern called a furin cleavage site that made the virus much more effective. This furin cleavage site was not part of any other human coronavirus and was certainly not a part of the original epidemic in 2003. In his search to understand exactly what it was and how it managed to be there, he stumbled on to the fact that the exact sequence of this furin cleavage site was also in a patent. Yes, the exact sequence had been patented a few years before. What would you do with that information? So, yeah, so that is essentially really what I knew from way back when. And so, what then happens is that this research eventually comes out. And this is therefore what the response to the research was. So, let me just reiterate it. Essentially, the scientist, he was working in the lab and he thought he would help by trying to find a parallel to the sequence on the spike protein of the virus because they're trying to work out where it's from and so on. So other scientists did this across in different parts of the world. I think Professor Petrovsky was doing it in Australia. This gentleman was doing it in the USA. And when he did the sequence, he found an unusual sequence that's a furin cleavage site, and this unusual sequence, which is not normally part of a coronavirus, and he did a blast search. This is just a kind of search where you put in the sequence and you look for parallels, you see if anything comes up. And what came up was a patent, that this exact sequence or the reverse of it was patented. And so immediately he knew this was a problem. And more than just taking the story, which is actually right here, this is the story here. The study that he did doesn't prove that Moderna created the virus. Because the furin cleavage site, that patent was registered to Moderna in 2016. And it went on to point out that the AP's assessment is that people claim Moderna created the virus. I don't think that's true. And their AP assessment is they found that a years old, well, about 3 years old, Moderna patent included a genomic sequence that was a reverse complement of a small portion of the SARS-CoV-2 genome. Experts say that finding doesn't say anything about the origin of the coronavirus. Now, I mean, it's one thing to say it doesn't say anything. I mean, it doesn't mean that Moderna is involved, but it certainly points to the fact that it's most likely a lab main virus. And what they worked out when they did it, because they weren't pointing any fingers. They were just trying to figure out how did this sequence get in the spike protein. And what they were interested in is that they usually you use that patent, that sequence, that furin cleavage site to assist a virus to get inside a cell. So this is in terms of cancer therapeutics. When you're trying to get cancer therapeutics inside a cell, you would attach a furin cleavage site onto the viral entry protein to aid it getting inside the target cell and delivering the package. And so what they were thinking was maybe somebody was trying to do this and it leaked. So they were thinking about just a lab leak. They weren't actually looking at who specifically was the cause of it. So it's a very important differentiation. It's just not what they were looking at. Now, they are talking about the claims being true or false. I am reading to you this here, and you can see, let me add it to the stage again. And so they went on to say that, but experts and the author of the study say that the paper does not prove that. They're very clear about that. He said, they quoted, "We understood from the beginning that this match could be a random chance and stated this in the article prominently." This is from Dr. or Professor Bala Ambati, the study author who is an ophthalmologist and a professor at the University of Oregon. And he said they were not casting aspersions on any person, company, or country. They were just doing the research, as I said. So, as usual, I don't just take the story. I want a little bit more information. So, what do? I go and interview the person. So, listen to this clip from this interview here.
Is about 7 or 8 codons, so about 24, 26 letters. Is that present in the previous database of viruses developed in Wuhan? And it was not. But it was present in this 19 nucleotide stretch, was present in this patent in 2016.
No, hold on there a second. So when you saw that, what did you think?
Well, yeah, probably not rated G thoughts. So, when we saw that, first we wanted to confirm, is that real? And we did. And second, we wanted to learn more about what this patent was about. And it was a tumor biology patent. It's a tumor vaccine patent. Basically, it is a library of different sequences that were patented for proprietary tumor vaccines back in 2016. So, a very legitimate scientific purpose. When we first came across this, we were wondering what to do with this information. To be blunt, the summer of 2020 was a very challenging summer in so many respects, right? Nobody in our group wanted to publicize this, and we sat on it probably for um, 7 or 8 months.
Very important clip there because he's highlighting the fact that they didn't know what to do with this information. This was from summer of 2020. They saw this and thought, oh my God, what do we do? And so what he did is then he got a consortium of scientists, put the information there, everybody looks at it, and then they published, did a publication eventually where they didn't put a cause or blame, they just gave their analysis of it. And critically, what they said is that the chances of it occurring naturally were like 1,000,000,000,000 to 1. So that strongly supports the fact that this is lab leak. Okay. Now, again, people may be arguing about, well, what's the relevance? So what? Well, this is one where I think if you can't handle the conspiracy question, so from this point on, I'm just going to share with you my thoughts. This is the time where you can step away. Okay. So you don't have to hear my conspiracy thoughts. I'll pause 1 second. So here we go. What was very strange about this virus was the targeting. So, what I mean by that is this virus was exceptionally well targeted to older people and people with comorbidities. Now, in most viral infections, you have a U shaped curve, meaning that you have deaths at the very young and deaths at the very old because they are most vulnerable. What was extremely unusual in this case was that it didn't really kill neonates. Now, neonates don't have a strong immune system, which is why they're very vulnerable to ongoing infections, viral infections going around. And so you usually are very worried about neonates with poor immune systems. And if there's a massive viral outbreak, so if we did have another flu epidemic, you would find that the very young would be dying as well as the very old with poor immune systems. So what that indicates to me is that the it's not clearly somebody just playing around in a lab. This was very targeted, and my research in autoimmunity highlighted that the most important piece of the puzzle was serum ACE2. ACE2 is the entry receptor for the virus, the main one. And in people who are older, people with comorbidities, diabetes, hypertension, obesity, their levels of free floating ACE2 is higher than the normal population. And that was our research into autoimmunity. We said this is an autoimmune response for severe COVID 19 because the free floating ACE2 would bind to the spike protein when the virus gets in the bloodstream. It's not complicated. And all of the autoimmune responses we see fits. So, what it would therefore mean is that to be blunt, the target was older people, probably more so than the people with comorbidities. So now you have to step back and think, well, why would older people be targeted in this way? Well, there are a couple of things that I think are very relevant. One of them is that I don't think the Western world can afford pensions anymore. It is unsustainable. And the only way to cope with that is if you had a much smaller cohort of people who needed pensions. Because you remember, people are retiring at 60 and living until 82. When pensions were designed, it was for people to retire at 60 and die at 63, the coal miners and so on. And so there was no anticipation that people would live 20 to 25 years beyond retirement. And so there isn't the way to fund this long term. I think that's my opinion, and I think that is built out in terms of some of the economic models. The other issue is to do with frailty and dementia, conditions that at the moment they don't have any good answers for. Now, this one comes close to my heart because my research has been for like 12 years into dementia and trying to reverse it. Because I've always recognized that risk that if the pressure on the system became so great because of frailty and dementia, populations may therefore kind of accept that they can't afford them and get rid of them. And this, truthfully, is what we saw happen in the thinking early on in the pandemic, where there was a willingness to let the older, more frail cohorts go in preference for younger, fitter, working age people. So when you look at that, you realize that actually not only was this targeted, but I think the world and society are prepared psychologically for this outcome. So, this is why I say this is not over. So, when you look at what has therefore happened, because you don't have to just extrapolate your thinking, if this was a lab leak, okay, and the spike protein was specifically designed, you then take that same spike protein and you then give it to the population. As far as I'm concerned, that's a recipe for a disaster because you don't even fully understand exactly what was built into a spike protein. If it was built in a lab, you don't know the mechanism, you don't know what they were trying to do. Remember, this spike protein, it infects lymphocytes, it gets inside bacteria, makes them produce toxin, does all kinds of stuff that we've never seen before. So this is where now you have to be prepared. And what I'm saying is that this is not relevant to everybody. So when I say that, what I mean is that a big proportion of the population is younger, good immune systems, and people keep on saying, yeah, I'm fine, I'm not worried. Who you have to worry about is not you. Who you have to worry about is if you have any older loved ones. Because the implications of ongoing viral circulation, it's still going to be targeting ACE2. Therefore, the cohorts with elevated levels are going to be at highest risk for longer term complications. In effect, this is described as a slow kill, meaning that you will not see the manifestations of this in the short term. So, this is why I've been monitoring excess deaths, because based on the science, it has to rise. And actually, it is already up because it should be down. And so I'm anticipating this continuous rise in excess deaths. There will be collateral damage because there's some people who are more vulnerable to the mechanisms of severe disease around the long COVID type of patterns. And that's probably just the reality as to where we are. So when I speak about it, I am looking at it from a very broad perspective with the understanding that I have been thinking about what were the intentions with a lab created virus that was so targeted to older people and comorbidities. And if you want to understand my thinking, that's where I'm coming from. And so this is part of the reason why when I make the warning about what's coming, it's based on the fact that I know that the problem or the mechanism is still in place. It still has to fulfill the intention, unless you can find a way to mitigate it. And at the moment, because everybody is caught up in narratives, so you have some people who are pro-vax and they're just want everybody to be vaccinated. Then you have another group who are anti-vax and they don't want anybody to be vaccinated, and the two groups are arguing with each other. That suits this perfectly because it means you are not focused on what the real target is. That's the reality. That's my thought. And there is nothing I have to back that up. So if somebody's asking me for peer reviewed evidence as to why I think that, no, it's just When I looked at the evidence and I looked at the patterns and I looked at what happened in the pandemic and I looked at how things were approached and I reflected on the fact that when people like Dr. Shetty could have saved all these lives, that was ignored. Instead of him being, raised up and cheered, he is being hammered. Anybody who tried to do anything different was stopped. Any drug that could work that would probably mitigate deaths is stopped. The whole thing, to me, just doesn't make any sense. That's me from a conspiratorial point of view. And so, yes, when it comes to this perspective, I've had a lot of time to reflect on it. I've had a lot of time to look at the science. And critically, I haven't given up on trying to find ways to mitigate it. I don't think it's necessarily the right thing for us to just give up on people. We should always try to find a way to make things better. We can't necessarily stop everything from going wrong, but if we can save lives, I would think that it's worth it. And even in the elderly, who may have dementia and otherwise, if we don't find solutions for those conditions by addressing the challenges today, we will still have to face them in the future. Thank you very much for listening. I hope you found that valuable.